Does this work? Okay. My privilege to be able to introduce Darrell Kotton, today's Green Round speaker. I'm going to have to ad-lib it. I was going to print out his CD to help me, but I didn't have enough paper in my printer, and the tray was full. Darrell went to attended college at the University of Pennsylvania. Got his master's degree at the Berkeley School of Medicine, Berkeley School of Music here in Boston. Then attended medical school at Washington University in St. Louis, and then went back to Hospital of University of Pennsylvania for his medical house officer training. We were thrilled to have the luck to match him as part of a geographic couples match with Darrell and the other Dr. Kotton, his wife, when they came to Boston in 1998. After his clinical year of fellowship, Darrell spent his first research postdoctoral fellowship with Alan Fine in the laboratory and in the pulmonary medicine section. Made the sentinel observation that alveolar macrophages are long-lived and perhaps may live for the length of the life of many mammals. That was an interesting observation and an important one because it highlighted the fact that they might be a target for gene therapy. And that was what Darrell's original interest was. As a result of that, he did a second postdoctoral fellowship with Richard Mulligan, in which he learned the techniques and tools of manipulating viruses for gene therapy. When he came back to Boston University, he brought with him Gustavo Moltosowski and George Murphy, who went to the GI section and the hematology section. And they began to do some interesting studies related to gene therapy when Yamanaka's an important observation that you could convert somatic cells into pluripotential stem cells. And that set upon their entire next phase of all of their careers, and particularly Darrell's, in identifying ways in which he could create and utilize inducible pluripotential stem cells and use them as a model for human lung development and, in addition, thyroid development along the way. So a few years later, Darrell and Gustavo and George started the Center for Regenerative Medicine, and that will be the topic of Darrell's talk today and its 10th anniversary of its opening. And he'll give you some highlights of some of the extraordinary progress and discoveries that they've made over the last decade. Decades ago, it was science fiction that you could convert somatic cells into stem cells and then redifferentiate them into all the organs of the body. And today that's real and in science. And Darrell will tell you a little bit more about how we can use those tools to repopulate an injured lung. So it's my pleasure today to introduce Darrell Kotton, our lecturer. Thanks.
Can you hear me okay? Yes, okay, great. Well, thank you. I know Friday at noon is a busy time, and I appreciate your attendance. I've tried to prepare a talk for everyone and highlight, because it is our 10th anniversary of the move of the CReM into our physical center, I want to highlight some of the accomplishments of my colleagues and give you a story of what our journey's been like. To change things up further, I usually run out of time thanking people at the end, so I'm going to start with the thank yous, because when we reach 10-year anniversaries, we're, I think, unusually thoughtful about looking back and thanking those who came before. So let me first thank our mentors and those who came before. Alan, I arrived at BU in 1998 as a pulmonary fellow with the interest to be 100% clinician. It was Alan Fine and his lab that really turned me on to science and changed my career trajectory, and I'm really grateful to him. Richard Mulligan was my postdoctoral fellowship mentor at Harvard, and it's there. I'm grateful to Richard for all he taught me, and I'm especially grateful for bringing together Gustavo, George, and I, who were postdocs at the same time in that lab. And this is really where we in that lab is where we hatched these plans to develop a collaborative unit, now a center, and so I'll tell you about that story. There were amazing scientists and mentors along the way. Gordon Keller, who developed stem cell directed differentiation. And right here in the Pulmonary Center, we have amazing mentors in David Center. Thank you, David, our chief. Jerry Brody, who died this past year, who we miss greatly. Mary Williams and Jay Misgard, all of whom are exceptional scientists that really inspire us. We should remember our chief, Dave Coleman, who really was a champion of launching the center and funded it, and Katja Ravid, who awarded us an ARC as part of her new Evans Center that helped us thrive. Karen Antman and Kate Walsh brought together BU and BMC to co-fund a center that has a physical space that is one of the few examples of joint BU-BMC shared space on the campus, and we're really proud of that history and thank them. And of course, I want to thank David Selden, who's no longer with us, for whom my chair is named. I'm fortunate to have a chair named after someone that I know and loved and inspired me, and so I really hope this does justice to his legacy. Of course, our CReM PIs, there are 9 of us photographed up there. Thank you, Gustavo, George, Andrew, Valerie, Finn, Angie, Kim, and Kostas, and our 2 PIs who were recruited away, Jason and Laertes. And we wouldn't function without our CReM managers, Meenakshi, Gabby, Greg, Marianne, and Pushpinder. And so keeping in mind the memories of David and Jerry, let's launch into, this is for the house staff now. Think about the patients. Let's think about the amazing need we have to better the suffering of our patients. We have a lot of answers. For example, consider these radiographs. We know what causes these problems, and how are we going to fix them? This is what patients are asking us. These 2 questions I'm giving you below to kind of inspire the house staff to think about how we start to answer very difficult questions. The X-ray on the left is bronchiectasis from cystic fibrosis. In the middle, bronchiectasis from primary ciliary dyskinesia. On the right, a CAT scan with a severe interstitial lung disease in a child, which is unusual. And in each case, we know the cause. It's a single gene defect on the left, CFTR mutation, the middle, DNA H5 mutation, on the right, a surfactant protein C mutation. And each of these mutations is in an epithelial cell, lining cell of either the airways on the 2 on the left, the alveoli type 2 pneumocyte on the right. So we know the cause, and the questions, therefore, are difficult. The patients, residents, are they asking me this? If you know the genetic cause, where's our cell therapy? Where's our gene therapy? We've heard of CRISPR. Where is it? Why can't you cure this disease? They tell you, my friend paid $10,000 out of pocket in a clinic in Florida and convinced they felt better and less short of breath. They had stem cells. Shouldn't I get that treatment? What would you say? What will you tell them? So by the end of 60 minutes, I hope you'll be well armed with some answers to those questions as we address the science. And the following is the first answer I give them, which is help is on the way. And these 66 incredible young scientists who are committed to answering those questions and bringing to fruition cell and gene therapies for those patients. So help is on the way is a really important answer. In the last decade, we formed a physical center where these people live on a single live and work. They live at homes too, by the way, but they spend some time in the center on one single floor, 670 Albany Street. On the second floor, you may know it better, is the pathology building. And there we eat, breathe, sleep, drink science. We have 66 people, 13 faculty, 9 independent PI-led research groups across disciplines and divisions and sections. We are 2 institutions, BU and BMC, in the same space. We have one mission, advancing science to heal the world. And this is some of the highlights of the last decade. So the talk will cover basically 3 epochs that I want to summarize in our existence. The first is committed to reprogramming to make stem cells from our patients, iPS cells, explain what they are. We'll move on to directed differentiation, how to coax those cells to desired cells, organoids, tissues. And lastly, how to deliver them into the body, engraftment, the new horizon for cell and gene therapies. It is the answer to the question the patient was asking us. How close are we to clinical realization of stem cell therapies? I hope I'll let you leave the room with a better answer than you have now to that difficult question. So as we embark, we come to a grainy old schematic. The background is blue. Remember PowerPoint blue backgrounds when we launched? This is a slide made by Gustavo, somewhat whimsical. He's got Albert Einstein on the top, but it shows the vision what we're trying to accomplish to answer the patient's questions. The idea is you take a patient, take their somatic cells that are easy to access, a drop of blood, a skin biopsy for fibroblasts, reprogram them into the exact equivalent of embryonic stem cells, a pluripotent cell in a dish that can make anything. That was the goal. Differentiate them. How? We didn't know, but we were going to try to differentiate them to the desired lineages, model their diseases, and put them back in the body. So this schema that Gustavo made when we launched, I can't emphasize enough, we didn't know how to make the cells. We didn't know what the cells were. We certainly didn't know how to differentiate them, although we had ideas that we'd follow developmental biology, which we didn't understand. We didn't know the tools, the signals, the pathways, the cells. We would need to modulate DNA. This technology had not been invented yet. CRISPR did not exist. We would need to profile the cells we were going to make. Single cell technologies, nobody dreamed of single cell RNA-seq in those days. We didn't know how that was going to be done. We need to condition recipient tissues, deliver the cells with new technologies. None of that we knew how to do, but we had faith that somehow with science and colleagues and working in a community, we could make our way around the circle. So I will cover what is now a revised, white background version made by Joe Kaiserman and Andrew Wilson. Thank you for the slide. This is the same, but now schematized with a little more polish. Reprogramming will be the first part of the talk of how to make these induced pluripotent stem cells in the laboratory and the difficult work of differentiating them to the cell type of interest. We won't spend time on the drug precision medicine prediction models and disease modeling that are a big part of our center, and I've spoken about those on previous grand rounds. We'll focus instead on the new, exciting, rapidly emerging approaches for delivering them into the body through transplantation to regenerate in vivo organ function. Okay, let's get started in the first part of the talk. Reprogramming, a trip down memory lane. If it's around 2006 to 2008, Gustavo Masoslavsky has this idea to take the brand new reprogramming technology, which involves 4 transcription factors put into a somatic cell, like a blood cell or a fibroblast, and he combines these into a single vector called the stem cell cassette. This is actually the map of his idea for genes in one cassette that's excisable, and we go to work reprogramming. We're very excited about this vector and its name. Gustavo is buying a bumper, a car license plate that has the word on it. Everything we do is Stemka, Stemka, and he wins the Innovator of the Year award for B in 2017 for this invention, rightly so. So why the excitement? What does this let us do? So let's just go through a reprogramming experiment. Take a fibroblast from a mouse tail tip. Very important that we start with mice. Doctors, interns, residents, mice are very important. We work this out in mice, and then it works the same in human. Let me say it again. It just works the same in humans and patients. Start with the mouse. This is what happens. You take tail tips of a mouse and grow fibroblasts out the same way you would from a patient's skin. You put in Gustavo's Stemka. Some cells round up and they start growing against the lawn of non-reprogrammed cells. This is called reprogramming. It's a change in the program of the cells. The chromatin is different. Everything is different, but it's recognizable. It looks like an embryonic stem cell in chromatin, in profile, molecularly, phenotypically, its potency. What it can do is like an embryonic stem cell. It's pluripotent and can make anything, but it's named, not embryonic stem cell. It's not from an embryo, it's from an adult mouse or adult human. It's named induced pluripotent stem cell, iPSC. You'll hear this over and over in the talk. This cell can now be picked up and passaged infinitely into new wells and shipped around the world for biomedical research. It's very important. It's a stem cell because it can differentiate, but it can also self-renew indefinitely, essentially without limit. Okay, so these are the stem cells from a mouse. How do we know they're pluripotent? Is it because I told you? No, it's because we can do experiments like this that are very easy to communicate and understand. We're looking at a blastocyst embryo from a mouse that is in a white albino mouse. There's a suction catheter. Here comes an capillary tube. It injects 40 or so cells from the reprogrammed tail tip fibroblasts that we think are pluripotent stem cells. If they behave like ES cells, they should chimerize. They're taken from a black mouse so we can track them. It should chimerize in this developing embryo from a white mouse. We can put this embryo into a foster mouse mother. 19 days later, pups are born. We simply ask, what is the coat color of those pups? And they look like this. They're chimeric mice where the black coat colors contributed from the reprogrammed cells. So this is proof of of pluripotency and developmental competence, but the cells can also contribute to the lungs and the liver and the intestine and even the germline. And to test for that possibility, we breed those chimeric mice with an albino mate and look at the next generation, which is black. This is called germline transmission. It's the highest level of proof, functional proof of pluripotency induced pluripotent stem cells. So this is an exciting picture. This is an exciting moment where George, Gustavo, and I would be calling our friends, our families, sky-fiving, committing our lives, our short lives, to trying to do this that happened in the mouse from humans in a laboratory dish and put it back in patients one day. And that is the birth of the CReM. That idea of going around the circle that we knew was possible from this photo, we're going to try to accomplish now. That was mice. When it works in mice, it works in humans. This is human. This is a human embryonic stem cell-like colony, not made from embryonic stem cells, but made from a little boy's skin fibroblast. The boy has a lung disease. We take a sample of the skin, reprogram the same way with the same factors, the same vector, STEMCA, other vectors we now use that might be a clinical grade. We have a lot of options. The result is no longer a fibroblast, but a colony of pluripotent stem cells imbued with the same competence as ES cells. Now, this is made from a patient that has deep clinical phenotyping. We know the clinical phenotype. We also know, of course, that each cell carries the entire genetic background of that patient, including the mutation. And we can now gene edit that mutation with CRISPR technologies that were discovered along the way. So that's one child with one mutation causing one lung disease, but you can do this over and over for all the diseases we're interested in at Boston Medical Center in the CReM. So we go to our clinics, consent patients. We don't skin biopsy them anymore. We take a drop of blood. We use the same reprogramming I've just explained to you, and we make vast banks of hundreds of cells from hundreds of patients that we then self-renew without limit. They're not precious. I mean, they're precious, but they're not limited. They can be shared around the world, and that's what we do. We share these cells, and we study them in the stories I'm about to tell you. Okay, so let's get back to the patients, the 3 X-rays. We're interested in understanding their diseases and helping them. We have made progress over many years in reprogramming their cells. So these patients and others like them with their mutations now are in the bank. Their iPS cells reprogrammed from just a drop of their blood are in the bank, ready for biomedical research. This is a big, big step here at Boston Medical Center. Okay, so what are we going to do with the cells in the bank aren't worth much if we can't do something with them? Well, the first thing to do is we have to turn them into the cell type of interest responsible for this disease. These are lung epithelial mutations, remember. So back to our schematic. The second portion of the talk now is to try to take those cells in the bank, in these 3 patients, from the child with the CAT scan I showed you, the NKX2.1 mutation, and differentiate them into the cell type of interest, lung epithelium, airway epithelium, type 2 pneumocytes. How are we going to do that before we get around the circle? This is a really vexing problem. The way you do this is you look to developmental biology, which I find very humorous because I skipped those lectures in med school. I found them irrelevant and boring and uninteresting. And now it's all I read. It's what I love. It's the Holy Grail. It is the roadmap of how we do our work. This is a mouse lung. It's taken 13 days into development. We've targeted it by putting a GFP reporter, green fluorescent protein, right into the NKX2.1 locus. If this locus comes on, it tells us the cell has decided to fate, to lung epithelial fate and nothing else forever and ever. It will always stay on in that lung as long as it's normal. And we make movies like this. Every 3 hours, Ian Kinslinger, our postdoc, is filming this lung branching and budding in the dish. It's beautiful. We need to understand these programs. What is regulating these fates? How are the cells branching and differentiating? Where did they come from? Where did the cells originate from? What is the moment the GFP reporter turned on? These are the questions that we obsess over, because figuring out that roadmap and those secrets then guides how we handle the iPS cells in the laboratory dish by recapitulating this same process. So where did these cells come from? Thank you, Finn Hawkins has schematized it for us here. If you're in the blastocyst embryo in the pluripotent cells, that's the inner cell mass, you will go through gastrulation to form the 3 germ layers, including our precursor germ layer for lung epithelium, that's definitive endoderm with the gut tube here. And when we reach the stage where this locus comes on NKX2.1, there it is in green in the mouse, it will thereafter stay on, and the branching differentiation program gives rise to the entire lung epithelium, airway, and alveoli. Now, if we're going to do this in a dish, we're going to need gene markers because we're not going to have this histology and anatomy. And here are the names of gene markers we follow, like road signs, as we culture cells in a dish. Now, if we get all the way here to making a lung cell, we're only halfway done. And there are a lot of pitfalls. That gene is also expressed, not many other places, but expressed in the thyroid, expressed in the ventral forebrain. This is not going to be easy, but it's going to be easier to make those other lineages that we're not trying to make because they evolved earlier in evolution and they express the same gene if we're just looking for genes and green cells. Okay, so if we get, despite those exits from the highway we're on, if we reach NKX 2.1, we're only really halfway done. We have a long way to go from this primordial progenitor to pattern the cells into airway, proximally, distal, alveoli. The airway becomes secretory, ciliated, basal cells. The distal ones go into the type 1, type 2 pneumocytes. This is like all of lung development in 30 seconds. It's hard to digest, but it's the most critical thing we do is understanding how this works. When we understand it, we've spent a lifetime, 20 years or so in my lab and a community and the same stories repeated in all the CReM labs to generate maps like this. It's like recipes. You see growth factors that we add in sequence at different stages to our iPS cells in the laboratory dish. It coaxes those cells to the next step in gastrulation, definitive endoderm, foregut, endodermal patterning, the NKX 2.1 positive lung epithelial progenitor. We obsess over the signaling pathway Wnt. We turn it up high, it makes pneumocytes. We turn it down low, it forms airway. Okay, so this is a lot of word salad, genes, recipes. And like cooking, a recipe can be boring when you just read it on a page, but it's amazingly fun and interesting when you actually cook, as we do. So let's take a little path down. You know, this makes it look very clean and nice, but let's memory lane. Let's go through endoderm, and foregut endoderm, and add some factors, a whole cocktail, because we didn't know what these factors were when we started, and take a little movie of the GFP gene coming on in the NKX 2.1 locus in mouse, embryonic stem cells that are differentiating. Here's the cocktail. We know we're on the right track because we can get green cells. And we watch in the laboratory, here's some dark cells, and like popcorn, one turns green right here. Yeah, and then there's a whole field of green cells. We are excited back in 2008 to 2012 making this movie. We're excited because we have some lung cells. But when we profile the cells, remember this gene is also in the thyroid. Some of them are thyroid cells. And I'm a pulmonary critical care doc, and I can tell you that critical care docs are tough. They've survived the pandemic. But if you want to see a critical care doc humbled, show them a set of thyroid function tests. They will melt. This is very intimidating. We call everything euthyroid sick. We don't understand any of it. But we do know when we see thyroid genes here that we're in trouble and we need help. And we do the same thing we do in the ICU, which is we call an endocrinologist. That's all there is to do. So that brings me to a friendship and long-lasting collaboration with Tony Hollenberg. At this picture, he was the endocrine chief at Beth Israel. And we write grants together, and we wrote this paper together. So serendipity, on the way to a lung, funny things happen, and we don't view those as failures. We listen to the cells. We listen to the data. Thyroid specification and what growth factors to add to encode thyroid fate was totally unknown. Totally unknown when we did this work. We stumbled on the factors. Here they are, BMP4 and FGF2, that stimulates those cells to become thyroid. You'll notice those are only 2 of the cocktail here. So that took us a while to figure out which of these actually made the thyroid, and then we could make it in pure form. Amazingly, those cells transplanted into hypothyroid mice form functioning thyroid follicles and rescue the mouse for a long time. This might be a future treatment for hypothyroidism. We have grants, Tony and I, to work on this. He's our chief, but he's still working on this. And we're in hot pursuit of treating hypothyroidism this way. And I always think when I present this of our first author, Anita Kerman, who was tragically killed right when this paper was published. And I miss her and I remember her when I show this kind of work, and let it inspire everyone in this room to know that when you ask questions and have some answers and share it with the world, you get to live forever in a way. This is Anita's legacy here. Okay, so if we figured out which factors make thyroid, We also know that the others in the cocktail probably make lung, and in fact, that's what happens. And here's a fast forward with several papers that now delineate this recipe in much more simple form. Take a gut endodermal cell, expose it to extremely simple conditions, just 2 growth factors, it'll fade to thyroid, NKX2.1 positive thyroid. Expose them to Winton BMP with some retinoic acid, it'll fate them to lung primordium. So we're halfway there, and we can make pure populations separately, which is very different than in the movie I showed you. So let's keep showing movies to tell the story. We're showing a mouse movie. Now let's go to a human movie. This is the work of Finn Hawkins. Now we have the roadmap partially there to get to lung primordium, and Finn Hawkins goes to work, but we're now in human cells. So we're now into patients. We take a cystic fibrosis patients, a little boy's fibroblasts, reprogramming them with the Stemka. It's an iPS cell that can be gene edited or not. We gene edit to put the same green fluorescent protein right into the human genome, something we never dreamed was possible until gene editing technology was invented right in the middle of us doing our work. Here is Finn's movie. The cells are human, cystic fibrosis iPS differentiated a long way, all the way through gastrulation into endodermal foregut cells. The GFP gene is in that NKX2.1 locus, and they're being exposed to those same factors, Winton BMP, that we figured out in that mouse system. Now, this is the cystic fibrosis cells having some cells turning green in response to these factors. These cells turning green is an amazing moment to us. We call it the Big Bang of lung development. It's the moment a foregut cell specifies to lung epithelial cell fate forever thereafter. That is what we think we're visualizing with this kind of movie. It's just one gene, so we have to profile all the genes. We can take the system that's somewhat chaotic. I know you think it doesn't look like a lung, but if you purify the cells, you can replate them into these beautiful spheroids. They're like a soccer ball in 3D matrigel, a single layer thick epithelium that's cuboidal. All the cells maintaining GFP in this experiment done by Finn. It looks a little more like a developing lung. When we build sandwiches, we call recombinants of bringing this pure epithelium together with the lung mesenchyme. And that looks like this, with human NKX in the middle, stained, and a mouse mesenchyme wrapped around it, differentiating, cross-talking with these cells. Now, we're only halfway there with the lung primordium, so we've got to get all the way to the end. And we spent many years going to the first end, which is a type 2 pneumocyte shown here. This is an electron micrograph, the edge of the sphere. When the cells stay in high wind, go all the way to a type 2 pneumocyte. They will form apical-basal polarized cells. There's a tight junction here. These are lamellar bodies packaging surfactant that get dumped into the lumen of the sphere on the inside. So these are surfactant secreting. These cells, when made from children with that surfactant mutation I showed you in the CAT scan, will model that child's disease, will model the response to the drugs, the new drugs we're trying to discover to help that child. These are very powerful disease models and precision drug therapy predictors, which I won't talk about because I want to move on to the engraftment. Okay, we've talked about reprogramming, the first epoch of the CReM, I call it. We talk about directed differentiation, the second epoch of the CReM. I'm just giving you an example of lung. It's the same if you show up in the Murphy lab and want to make blood, or the Gruen-Evans lab and want to make liver, or the Mostoslavsky lab and want to make intestine. The principles are the same. Okay, but we have a very hard task now. We're trying to deliver those cells into the body, and this will be extremely dangerous in humans. These cells could be cancerous in very quickly. So we want to start not in patients, but in mice. And this is the story of how we worked on cell-based therapy. This is a schematic of all those cell types of the airway epithelium of the lung and the distal alveolar epithelium. These are a lot of names and colors. This beautiful schematic made by Michael Harrigas is beautiful and intimidating. There are too many cell types to make. We try to make them all, but if we want to graft them all, where do we start? How long will they live? These are massive hurdles. So the philosophy here we came up with is the cells are not all equal. Some of them are stem cells. The green cell here is a basal cell of the airway. That is the airway stem cell from a huge literature worked out in mice. That cell will self-renew for life and differentiate into its progeny, which are secretory and ciliated cells. In the alveolus, the best studied progenitor is this type 2 pneumocyte, which can self-renew and differentiate. So the suggestion then from nature is you don't have to graft all the cells, but if we could graft the stem cell niche, a basal cell in its niche, a type 2 pneumocyte-like cell in its niche, perhaps those cells will then do the work for us of self-renewal and multipotent differentiation for life. That is the goal. So those are the two cells we focus on as part of this work. So here's the team airway meeting in the pandemic. Finn Hawkins is a team leader here, and we're brainstorming how are we going to get those cells into the airway. Finn, your eyes are still closed. I keep waiting for them to open. But this was the plan of our student, Martin Ma, graduate student, MD, PhD, who is summarizing here. This is the plan for regenerating the airway with the iPS-derived lung cells we've been generating in the dish. And we're going to do it in mice first, and then we'll see about humans later. We have a diseased airway, such as from the cystic fibrosis or primary ciliary dyskinesia patients, scans and x-rays I showed you in the first slide. That diseased patient has a gene defect in these cells. Martin is imagining that if he de-epithelializes the airway, he can make some space briefly to receive new engineered cells, especially if they've been gene edited. That would fix the gene defect. And then if we get the cells to stick, if he reconstitutes the stem cell compartment, the basal cell shown in green here at the bottom, the base, those cells will self-renew and differentiate. That's the plan. Make space, optimize the cell source. Basal cells is the source we're after. Graft the tissue stem cell compartment. That is the dream. Okay, so here goes Martin. He's giving a detergent, polydocanol. Over time, he's taking H&E stains of the mouse trachea. You can see the cells lifting up. They're severely injured from the detergent at 3 hours. By 5 hours, they're gone, and you have some round cells left with a lot of space, real estate in between. This is basal cells that then rapidly regenerate by 12 hours, so much proliferation. You've got a coating of basal-like cells, and by 2 weeks, the whole thing is regenerated essentially back to normal. So Martin thinks you have a narrow window, 5 hours, based on this. 5 hours, get the detergent in, and get the cells in, and see what happens. Okay, so here it comes. We have a basal cell. This is the cell source we're going to put in at 5 hours. This is a human basal cell developed through Finn Hawkins' lab and published in this cell stem cell paper in collaboration with Brian Davis's lab. If you make one basal cell from iPS cells in the dish, it's a magic cell. It is a stem cell. It will self-renew indefinitely, clonally forming these bronchospheres, as we call them. Here's an actual picture of the data. Green are the basal cells with this marker. Pink are the secretory cells, CC10, immunostain, and red are the multiciliated, waving ciliated cells. This is not mesenchyme or vasculature immune cells. It's an epithelial-only bronchosphere. We call it an organoid because it has some components of the bronchus. Okay, it goes over and over, self-renewing. You can passage it for a year, share it, ship it around the world, and we use profiling like single-cell-seq that was invented along the way to understand the whole transcriptome of every single one of these cells, and it looks like basal, secretory, and ciliated cells when we go through all the genes. That's a human cell, and Martin then adapts this protocol to make a mouse basal cell from ES cells or iPS cells. Why? Again, it's just dangerous. We want to start with mice, and the biology is the same. It will translate, we think, easily to humans once we work out the variables in the mouse. The key then is, and this is some phenotype, we don't just take pretty pictures. I guess I'm trying to say we do reverse transcriptase quantitative PCR, comparing the cell's expression programs to primary real lung cells like tracheal epithelium in the mouse. We use single-cell RNA sequencing. We use a lot of technologies that can be distracting in a talk trying to cover 10 years. So let's just stick to the pictures for a moment and say they look pretty. They're like soccer balls, single epithelial layer expressing markers of basal cells, keratin 5. And the goal then is to start to think about a preclinical simulation of a future therapy. You take your patient, take their iPS cells, we know how to reprogram, turn them into lung primordial epithelial cells, which I've been showing to you, push them all the way into a basal cell and see if you can get that basal cell into the recipient mouse trachea 5 hours exactly after the detergent makes space for the cells. So this is the Martin's plan. He's going to color them green or red with the lentivirus and put them intratracheally, one day with a bronchoscope, for now just squirted down the back of the throat into the lungs, and see if they graft in the trachea. Okay, with me? Want to take a look? You want to see how it works out? All right. Mouse trachea, taken out of the animal, fluorescent green channel of the microscope, no signal. There's no transplant here. If we repeat this with 7 million cells, no signal. Why? Because no space. Repeat this again with 6 to 7 million cells, 5 hours after polydocanol makes space, and you get a picture like this. The entire trachea has a very robust green fluorescent signal. What are these cells? Is this a reproducible signal? Here's a number of mice recipients imaged at up to hundreds of days after transplantation with these GFP-positive donor-derived cells that we engineered in culture, transplanted into that mouse recipient trachea. And we score it using fluorescence FACS scores, so we can quantify how many of the cells are donor-derived. It's the majority of the trachea, as you could imagine, looking at the pictures. And then let's take a look at the section and see where the cells are and what they might be. Here's an H&E stain. There's our old friend, the thyroid, with the follicles there. So this top of the trachea here, bottom of the trachea here. Here's the trachea of the mouse. And here is the fluorescence overlay. You can see the stripe doesn't go deep. It's just the single epithelial layer. It's not in the mesenchyme. It's not invading. And it seems like the majority of the epithelium is donor-derived. And this will be the same if we take this picture after a year. Same answer. What are they as we immunophenotype the cells? They are the basal cells, secretory cells, and ciliated cell phenotypes by immunophenotyping. For example, here's NCAGS 2.1 in the GFP-positive donor-derived cells. Here is this purple stain here surrounds these GFP-positive basal cells that are donor-derived, and the ciliated cells are GFP-positive as well. And this signal will remain stable. Here it is again in another mouse followed now for 1 year post-transplant. Now, a laboratory mouse will last for 2 years. And so Martin is interested if we go to the very end of the lifespan of the mouse, if we've really grafted the stem cell compartment, meaning the basal cell compartment of this mouse, the signal should not extinguish. It should be stable, self-renewing. That's what stem cells do for the life of the mouse. And he can come back then to 2 years at the end of the lifespan of a mouse that got the transplant of GFP-positive cell and take this amazing picture that was really celebrated in our center that day. He texted the photo to me. There were lots of celebrations. This is the evidence of grafting a stem cell compartment. So we have to do some homework and check some boxes here. So let's check the boxes. There's some functional assays that we need to do to see if the cells are really behaving themselves. This is a beautiful study done by Andrew Tilston-Lunell in Bob Varelis's lab. He's got the trachea fresh out of the mouse now, the recipient, hundreds of days out. He's going to video live cells. They're still alive. This is a GFP-positive donor-derived ciliated cell beating away. This is some secretory cells that are brighter. You can say that the grafted cells are functionally very similar to the endogenous cells. And here's another picture taken where we put beads on the trachea to see if the mucociliary escalator is intact in this highly grafted trachea. And these beads, look at these beads. They go, even though the trachea has been transplanted, they function by having coordinated beating that moves beads out. This is what happens in your lungs to move particulate matter out, let's say, for example. So the cells seem to be functioning well. And then in a really amazing experiment, let's just share with you a minute this experiment. Martin thinks that if the cells are really stem cells that we've grafted, basal cells, then they should self-renew indefinitely, maybe longer than the lifespan of a mouse. Now, if you're a blood researcher like Gustavo, George, and I trained as hematopoietic stem cell postdocs originally, there's a classical assay where you can replace a mouse's bone marrow with a hematopoietic stem cell. This is a myeloablated mouse. And you can take the marrow out of that recipient and do a secondary transplantation into another mouse. And you can keep that going up to 6 generations and no more for reasons that might relate to telomere length or other variables. So knowing that literature, we decide to try to repeat that experiment. We're going to take the donor-derived GFP-positive cells in this recipient's trachea and transplant it into a secondary polydocanol conditioned mouse and try to repeat that over several generations. And there's a culture period in between. It's not quite going from the mouse into the next mouse. It's going through culture to make more cells and into the next mouse. So how many generations can one go? This is, I think we're up to 7 now, Markin. This is the first, am I right? 7? Yeah, this is the first 5 generations out of 7 shown here that he published in his stem cell paper last month. And you can see the amazing signal that just keeps going now, I think longer than the lifespan of a mouse. You can have a donor-derived cell become a basal cell and then graft and survive and self-renew for even longer than the lifespan of that animal. It's just an amazing observation with sustained immunophenotyping and a nice, robust, sustained signal that statistically is undiminished in our hands. Okay, we've been talking about mice for a while. We're talking about grafting airway cells, and we need to be mindful that talking to patients about mice, it gets frustrating. This is the Alpha-1 Foundation's local support chapter that visits us all the time. We share this information with them. They have the most common genetic cause of emphysema. They want the cells developing fast, and they push us. Okay, where are the human cells? When are you going to get to humans? And we're grateful that they push us so much because the patients are waiting. And here's Andrew Wilson, who leads this incredible Alpha-1 center and brings these patients in and push us. So we tell them, and we want to push it anyway, that we will repeat this as the next preclinical step using human cells made from normal volunteers or patients that are iPS cells differentiated all the way to the basal cell. Now we take an immunodeficient mouse and using the same technique, polydocanol conditioning, transplant human iPS-derived basal cells into a recipient mouse trachea. And this is, they colored red now by a lentivirus. This was the inspirational picture the gang took during the pandemic. It was really a bright spot in a dark time. And to think that we're looking at human patient-derived iPS cells now gone all the way around the circle to in vivo engraftment in a recipient mouse's trachea was an amazing moment. It's amazing, but it also lets you ask some really interesting questions about deterministic fates. We know that mice and human are not identical. For example, there are goblet cells in human airways naturally, and they tend to be absent from mice except in certain asthma models. And when we do this transplant work, mouse into mouse, we don't see goblet cells as one would predict. But humans have goblet cells normally. And so the question was, what will these cells be? The human cells in a mouse trachea, would they be goblet, the human genome wins, or would they be not goblet, secretory cells, as in the mouse niche wins? And so we took a look at the immunophenotyping, and these are goblet cells stained MUC5B positive by Martin. They're GFP, now colored with a lenti-GFP virus. The human donor-derived cells in the mouse trachea turn into goblet cells, and we thought that was a really interesting answer to an interesting question that Martin published. And again, just to emphasize that the molecular phenotyping is key here. We don't just take the pictures, we also profile by single-cell RNA sequencing. And whether we use primary basal cells or engineered iPS-derived basal cells, the transcriptomes are amazingly similar across these different transplants. And the phenotyping of the cells is quite impressive for all these genes that validate that they're basal, secretory, and ciliated cells. Okay, so the airway is one part of the lung. We've all been through the pandemic, and we know that you're really in trouble if your alveolus is injured. So what about the alveolus? Taking these principles of making space, optimizing the cell source, and then grafting the stem cell compartment, let's see if we can get beyond the airway to reconstituting the alveolus. Alveolus, a much more complex structure. There's only two epithelial cell types there, type 2 cuboidal, type 1 flat, but the structure overall and the cells it's interacting with, very complex. It's very tough to make space there. So this is the work of Michael Harrigas. I'll just show a couple of slides. He published this last month in Cell Stem Cell back-to-back with Martin's paper if you want to look up more details. But Michael's idea was we're going to need to make a lot of cells for grafting the distal lung alveolus. And we're going to need to know medias, recipes for how to grow them. And he really liked this paper we found by Massimo Nishane that already developed a media that could grow out primary cells in mice to graft and successfully in that paper graft cells into the alveolus. So Michael wanted to engineer cells in the laboratory using these principles of directed differentiation, thinking that if you got the cell all the way to the right phenotype, as in this paper, a phenotype called a bud tip cell, that he could grow a lot of cells that he would need for transplantation. And this is his data that says the cells he engineered from embryonic stem cells grew just like the primary controls, undiminished over many passages, so he can make a lot of cells in this media. These are the pretty spheres of how he grows them, and then I think you're all waiting for the transplant data for the alveolus. So here's, and he uses single-cell RNA sequencing to characterize them extensively in that paper. Let's just do the transplant together. We're culling the cells GFP positive with a lentivirus. Michael's conditioning regimen to make space is bleomycin injury of the native epithelium in that mouse in the alveolus, and he's going to transplant in 600,000 or so cells that are lentivirally GFP tagged, and then look at the recipient. Now, these mice are not immunosuppressed. They are syngeneic, because this would mimic an autologous therapy in the future for a genetic alveolar disease, such as the SBC mutation in the child that affected the alveolus? Might it be used for IPF, for telomerase pathway deficiencies? Yes. Might it be used to reconstitute ARDS? More difficult, but you never know. This is the principles we're developing. Okay, take a look at this recipient lung tissue for GFP positive cells out after several weeks. And this is the immunophenotyping. You can see the mouse alveolar structure now, a lot of GFP signal. And when Michael puts on overlay of a type 1 pneumocyte protein stain, protoplanin, these flat red lines co-localized with these flat green lines, and these surfactant protein C and white dots in the cytoplasm lighting up their lamellar bodies in these cuboidal cells overlay with these green cuboidal cells. This is the overlay here. So this is the evidence that Michael successfully engrafted for many months durably a cell he had engineered that became a self-renewing type 2 pneumocyte in this recipient's mouse lungs that also had the capacity to differentiate and form the flat, what we've been thinking was a terminally differentiated flat alveolar cell. This is just mouse. This is not yet done with human cells, but this is the path we're on to try to form these therapies. Okay, so far I've covered our 3 epochs, reprogramming, directed differentiation, and gene editing to make cells, tissues, and organoids. We've covered engraftment with 2 example stories, and I'd like to close in the last few minutes with addressing this question of how close we are to clinical realization of stem cell therapies. This is the vision of DJ Wallman and Finn Hawkins. How are we getting to the clinics? Those basal cells generated from iPS cells, so impressive in the mice, have them all the way up to human induced basal cells living in the mouse trachea. Surely that must be a way to get that into our patients. So this is their vision. From bench to bedside, going through large animal models with ferrets, developing advanced bronchoscopic techniques to condition airways or treat them with gene editing tools, or one day get those same human cells that are living in the mouse into a CF, cystic fibrosis patient after editing, into a primary ciliary dyskinesia patient after editing. This is their vision. These are the steps being imagined along the way. And here are the grants that we're waiting for to support this. We have lung regeneration PPG that we've submitted with the excellent score, primary ciliary dyskinesia, team airway applications are going in. Anat Cohen has developed a microbronchoscope technique with mycoherigas where mice airways are getting bronchoscope to deliver these cells to try to work out all the techniques. Is this a pipe dream? Is this far-fetched? I think far-fetched is what we do in the CReM, and I say I bet they pull this off. I don't know if it'll be them or their trainees, but this is what we see as the path to the clinic for these cells. A special grant designed just for this is given by the Cystic Fibrosis Foundation. It's called the Pioneer Award, and I want to recognize our pioneer, Finn Hawkins, who we're all supporting as he prepares this application with this kind of incredible vision, which is just what we need at BU and BMC, I think. So I've been talking about the lungs for a while. We talked about reprogramming in the CReM in the beginning. Let's get back to the CReM. These are our 9 PIs. The story I've just told you is repeated over and over for each of them. Same concept, same approach, different diseases, different tissues, but same incredible stories. Let's just highlight some of them. The lineages being made that we want to study, disease model, develop drugs for, gene edit, and put back into patients are highlighted here across the CReM. The sickle cell amyloid program, making blood, making liver, with important projects designed to cure cirrhosis or understand alpha-1 antitrypsin deficiency, as led by Valerie Guan-Evans and Andrew Wilson. The cardiovascular lineages that I know many of you love, with amazing beating cells and endothelial and smooth muscle-lined vessels being generated. The other lung lineages of the vasculature immune lineages and mesenchyme are being generated, as are intestinal epithelia, many neuronal lineages, and of course, those endocrine organs, thyroid and parathyroid, all being produced in the center. What are some of the pretty pictures we've produced with some of these cells? Consider this. Valerie Guan-Evans' lab. This is Anna's work here, Anna Smith. These are human hepatocytes grafted into a mouse liver, occupying more than 30%. It's just a stunning graphic that just gives you hope that one day cirrhosis or genetic liver disease is going to have a better therapy thanks to Valerie's lab. The disease target they're in hot pursuit of is alpha-1 antitrypsin deficiency, and Andrew Wilson has the biggest bank in the world of iPS cells in the CReM from alpha-1 patients and runs an alpha-1 clinical center at BMC, which is one of the most renowned internationally. We're really so proud of Andrew. And here's George Murphy and Kim Vanuzzel's incredible Nature Communications paper asking the actual animal, the in vivo tissue, hematopoietic stem cells, what are their secrets in the programs that control their engraftment as a roadmap then to recapitulate that with engineered cells that can graft and maybe cure sickle cell disease one day. And then, of course, Angie Serrano, who has new zebrafish models. And I think if there's any message from these people, it's that translation is not a word that's just saved for some kind of science, that all the science is translational. All research is translational. And you can imagine the zebrafish, the mice, and all these models, how they're going to translate to the clinics. Okay, so a word about our patients then that are waiting with those questions I promised to answer. If you know the genetic cause, where are our cell and gene therapies? And my friend paid for stem cells, should I pay for them too? What will you say to these questions now that you have some knowledge? Well, consider that what I'm telling you is not just a futuristic, far off plan to put cells into patients. This is here and now. The clinical trials are here. They're here. Interns, residents, if you're on the Zoom, they're here. 102 pluripotent stem cell clinical trials already launched. Those we know some data from that are particularly well done and exciting, I'm featuring here. Okay, so they're not ready in the lungs yet, but they are being delivered. Human embryonic stem cell derived retinal pigmented epithelium already delivered into the retinas of patients with macular degeneration and published in 2014. Here's Dr. Takahashi announcing the first patient that has the same approach, but not from ES cells, from iPS cells, from their own iPS cells. And Doug Melton here holding a vial of cells. He's been working on how to make human embryonic stem cells, or iPS cells, into islet cells, into beta cells that can be transplanted. And they have been transplanted into insulin-dependent diabetics, one of whom came off insulin, right? This is just kind of safety trials, but there appears to be some efficacy. And it's just an extremely exciting time for diabetes patients and researchers like Doug Melton and the Vertex that's leading the trial here in 2022 and having the first data announced. Consider this. This is here and now. This year, Bayer Blue Rock announces their first results in 12 patients with human embryonic stem cell-derived dopaminergic neurons, phase 1. The early results indicate excellent tolerability, some hints of efficacy, but imagine people getting developmental biology understood of how you can go all the way from this pluripotent stem cell, all the way through ectoderm neural development into a dopamine-secreting, a dopaminergic neuron, surgically implanted in one side of the brain so you can compare the movement to the other side of the brain, and having 12 people already have results. It's just a time filled with hope. Unfortunately, it's also a time filled with warnings and shamans. Here's the answer to the patient's question: should you pay for the stem cells? I'm a stem cell biologist, stem cell researcher, and I can't keep up with the literature in the clinics. I don't know how any doctor can. So this is your armor. There's a website you can trust from our organization, the International Society of Stem Cell Research, ISSCR. They have designed a patient handbook on stem cell therapies that I refer patients to. We call this unproven stem cell therapies. I think that's a euphemism. I think the appropriate word is it's shamanism. Do not let your patients pay out of pocket for any stem cell therapy. They're not even stem cells. They're getting injected fat, bone marrow. Some of them are contaminated. They are not stem cells, and any stem cell given should be done for free in a clinical trial. It should never be paid out of pocket. That's the easy answer. Tell them to run away if people are charging them money, and refer them for self-education to these books. And the last time I gave grand rounds warning about this, I got hate mail afterwards from people who saw it on Zoom, and I welcome the debate. But that's what the science says today. Do not pay for unproven so-called stem cell therapies. Okay, in conclusion then, this is our summary in the graphic form. We told you about our vision, our mission, not knowing what cells, technologies, or approaches to use, and the amazing discoveries and 3 Nobel Prizes that happened along the way to help us do our work. In a community together, we figured out reprogramming with methods such as STEMCA and others in the community, how to differentiate the cells, to control fate by following the roadmap of the developing embryo, how important developmental biology research was to us and all we do. We didn't talk much about modeling diseases or testing drugs, but there are a lot of impressive papers from the CReM on that, and we spent a good deal of time with just a couple example stories of cell-based therapy, grafting the airway basal cell compartment, grafting the distal lung type 2 pneumocyte compartment. Let me then go back to the slide I started with. It just say journeys like this, they just aren't possible with those who come before and inspire us, whose memories stay with us, the people that make logistics work, and my colleagues, the 9 PIs at the Center for Regenerative Medicine. So help is on the way is the message I leave you with, and hopefully some interesting answers to the questions of our patients. Thank you so much for celebrating with us the 10-year anniversary of the CReM, and I'm happy to take all your questions. Thank you.
Darrell, a real tour de force. Congratulations to you and all the members of the CReM. Wonderful story. Scientific question. So, looking at the implantation of the basal cells into the airway epithelium and the tip cells into the alveoli, the basal cells then form this trilinear phenotype, and the tip cells go from type 1 alveoli to type 2, and then you can also make from type 2 that can go on and produce some type 1. What are the signals that are doing that inside?
Yes, what are the signals that control fate in situ? We read a lot of literature and study that, and some of those fate decisions you can control in vivo. I think a particularly delightful one that Claire Burgess worked on as a PhD student in our group, and it's just a really nice story. If you can engineer all the way to a human type 2 pneumocyte in a dish, so you're not in vivo now, that cell should be able to tell you the secrets of what changes it to a type 1 cell, and those should be the same that work in vivo. And that secret is it's hippo signaling. So if you activate YAP and TAZ and drive it to the nucleus, the type 2, its entire transcriptome changes to a type 1, and she can even get the beautiful flat shape. And there's many mouse genetic models that says that it works exactly the same way in vivo in mice tissue. So this is conserved from mice to humans. It's difficult to prove in humans with normal approaches, but with our approaches in the dish, I think in partnership with the in vivo mice, that's one answer to your questions. The answer about the airway is, I mean, there's a lot of literature about that similarly, and Wnt is a big part of that, and FGF signaling, and Finn Hawkins and his team have beautiful papers on also similarly taking a basal cell, modulating those signals to drive them to the other trilineage differentiation in the same biology that works in vivo. I hope that answered your question. That I'm just emphasizing that these are little vehicles in vitro that just open the book and let you reveal the in vivo biology and the signaling pathways, which are published from the group, actually. It's not just theory.
What trials are we going to be starting here?
Well, they're easier and harder. I think the delivering CRISPR editors and its base editors, prime editors into the body, I think Rick Ruber gave a delightful grand rounds and talked about some of the upcoming amyloid and other editing trials from companies that will launch starting in Europe are so exciting. And I think that should, in our lifetime here, while we're working at BMC, I hope that'll come into the clinics here on campus with editors being delivered to cure some of these genetic diseases. You could call it TTR amyloid. I think alpha-1 antitrypsin deficiencies being thought about by Andrew Wilson. And then these cell-based therapies, because they have such a different level of danger and difficulty with the conditioning regimens, I think that may take a few years longer. But I showed the slide in the airway because I particularly find the vision and the plan so compelling, and I hope we can do that on campus one day. Bronchoscopically delivered edited cells into cystic fibrosis and primary ciliary dyskinesia patients, I think may be possible in our lifetime. Ditto for sickle cell disease.
So the answer to the question to a patient, where should they go for gene therapy?
Yeah, yeah. I think go to the Murphy, Van Uytzel, and Hawkins and Wilson labs, and you'll be good. Yeah.
Any reason to bank really healthy humans, people who live a really long time?
Yes. I think this really highlights George Murphy's vision for aging, the aging research program, and his approach has been to make iPS cells and understand them when made from supercentenarians. So what encodes the secrets of extreme longevity? I think the same philosophy was saying these cells are little, microscopes and telescopes where you can look into the programs in a dish from those people who are living to more than 100 years old and figure out what encodes their longevity. And here's a nice publication in that area, and certainly more to come.
Thanks so much for that fascinating presentation. I'm wondering if someone bring that position.
Yes.
If you have any advice.
I have advice, yes. I'm a little bit old school. I think the trend is you're supposed to declare early and short track, and just, I think in kindergarten, sometimes we expect a physician scientist to know. So I didn't. I wanted to be 100% clinician. I'm a big advocate of late bloomers and preserving that people who are curious should be given protected time, resources to play. And there's no question too basic. And out of the most basic questions, like what makes the jellyfish glow green fluorescent protein, or how could you modify mRNA, comes life-saving therapies. And so supporting basic science, young people who are curious and decide late what to do is the key, and throwing them into fertile soil and environments like this, I think then nature takes its course, if you will. So my advice to young people is live a life of meaning and go for it. Don't be distracted by, the Snapchat and the Insta culture of happiness is the goal. Happiness is not the goal. The best way to happiness is to live a life of meaning that involves challenge and struggle towards a worthy goal. And with meaning comes extreme happiness. And watching Instagram, a TikTok video is meaningless, but it might make you briefly happy. We're not into making, I think, trainees happy. We're into providing them an experience that's extremely meaningful. That's my, I better stop there. Thank you for the question. Yeah. Thank you. Thank you. Is that okay? Thank you. Thank you.
Absolutely.
Thank you. Thank you.
Made science out of science.
And congratulations. Thank you.
You created something that